All right, can you see the presentation first page?
Yes.
Okay, good. So my name is Leho Tedersoo, and today I'm presenting the rationale and the principal or the preliminary results of the global soil fungal research project. So I will start with the Consortium project, and in the end I will talk about an additional endemicity study. So this research has no very specific funding, but it has been mixed from various sources, and of course all foreign contributors have also contributed from their own financial sources, either by arranging their collection trips or by covering some of the work and shipping costs. So if you have any specific questions, you can later contact me by my university email. And if you're interested to follow other research, you can sign up in Twitter and become a follower. So now to business. First, about the history of this global soil microbiome Mycobiome Consortium. We had this idea for quite a long time, and actually we started to collect the first soil samples in 2006. It was when we did our first expedition to the Seychelles Island. We focused mostly on mycorrhizal fungi on root tips and as fruit bodies, but we also collected some soils, although it was not very well systematized. In 2010, we thought that it might be feasible to do some more systematic global sampling, because the methods started to become available about massive analysis of soil biodiversity. And then we decided about this specific design of 40 subsamples from a quarter of a hectare. And at the same year, we started systematic collection of global samples, started in March 2010 from Madagascar, when we had one of the last African expeditions within my postdoc project, which was about African fungi. We invited many acquaintances, other researchers to participate, and within 3 or 4 years we managed to collect 365 samples, and 4 years later we were lucky to get this paper published in Science. Roughly at the same time with publication, there was a global mycological conference in Bangkok, Thailand, and there we had a meeting with many colleagues, and we decided to officially launch this global soil microbiome consortium, and our aim was to increase the sample size by an order of magnitude compared to what was previously achieved. So the next 6 years, we invited many people to join, I think we had this discussion with about 1,000 persons, and within 6 years we did active collection throughout the world. In the last, let's say, 12 months, we had very active molecular analysis going on. Despite the pandemics, we could do pretty well. And within this year, we started active data analysis and the process with preparing for publications. So finally, we managed to collect samples from more than 3,400 plots worldwide. It includes some of the plots that have been sampled more than once, so they are not all unique. And more than 120 people got involved. I must say it was extremely difficult to persuade people from certain countries to participate. For example, Indonesia was maybe the most difficult place to get samples from. And on the left, you can see this our sampling design, where red spots represent the 40 samples, and those tree-like figures, they represent woody plants. So this design applied to forest or tree-dominated ecosystems. Since you are not mostly molecular ecologists, I go through very briefly processing methods. So when we collected the soils, or our collaborators collected these were first dried and homogenized in our laboratory in Tartu. We performed DNA extraction from 2 grams of soil. It was a very slow process because it could be done only in batches of 6 or 12. For analysis, we selected the best potential marker for fungi, which includes the entire internal transcribed spacer region, plus a small end proportion of ribosomal small subunit, so-called V9 variable region. PCR was performed with extremely universal eukaryote primer, ETS9 MUN and ITS4 variant. We started with PacBio sequel 2 sequencing as soon as it came into the market, because it was the only cost-efficient method providing enough sequence data at that time. So the analysis was in a way waiting until specific or suitable technological advances. In the last few months, we have been very heavily working on bioinformatics, mostly done by Vladimir Mikrykov in my lab. This includes quality filtering, clustering at 98% to delimit species level taxa, and finally dataset polishing. At the moment, we are mostly progressing with data analysis, and we are preparing the data for databasing. It will be stored in various formats in the UNITE database, and also in the publisher's homepage. So this part of the data we are going to submit to the journal Scientific Data, and also publish analysis based on this data, all this our group together in hopefully as high-ranking journal as possible. So briefly about the results. So we were able to receive high-quality molecular data from 3,200 samples, which included almost 19 million reads, and these could be separated into 1.15 million OTUs, or roughly species level taxa. These more than 1 million OTUs included a bit more than 700,000 fungal taxa. And in the pie diagram on the right, you can see the distribution of taxa. You see that a bit more than 60% of the taxa belong to fungi, but a very significant proportion is also alveolates, animals, and amoebae from Rhizaria kingdom, but also a lot of vascular plants, bryophytes, and chlorophytes, which are in this light blue sector. Since some other databases use also the ITS2 region, and our original study in 2014 was also based on the ITS2 region, we sought to address whether this full ITS region is actually better. So we did some specific analysis, and we found that the full ITS has a greater species level resolution. It enables much stronger, higher level recognition of taxa. I mean here phylum and kingdom level identification and understanding what kind of organisms these are. Full ITS region also allows a more precise OTU calculation because it is not so sensitive to random mutations in the read compared to the short region. And we found that this V9 extra part of the small subunit ribosomal RNA gene allows us to resolve half of the unknown taxa that would not be able to be classified into kingdom or phylum level groups. The downside of full ITS region is, of course, additional chimera formation, simply because the region is longer and there are more places where the chimeric molecules could form. Nonetheless, we only found about 10% of chimeric molecules among the singletons, or those OTUs that are represented only by a single sequence, which shows that generally the quality of the reads is better than expected. When comparing the full ITS region to ITS2 region only, we found much higher representation of especially early diverging fungal groups in the full ITS data. And here in the pie diagrams, you can see that all from the top vertical line to this blue sector are ascomycetes and basidiomycetes, and the rest are early diverging fungal lineages or unknown groups, whereas this part is much lower when we look at the ITS2 sequences alone. We also sought to compare 3 different data sets based on this ITS2 region. In our data set, we found more than 400,000 fungal OTUs. In the global fungi data set, there were more than 900,000 fungal OTUs, and in INST and UNITE combined, there was a bit more than 100,000 fungal OTUs. So quite surprisingly, our dataset and the UNITE INST ITS2 dataset were more similar to each other than the global fungi dataset. Compared to any other datasets, this GSMC dataset has much greater proportion of early diverging fungal lineages. And for example, comparing to the global fungi dataset, many of the unicellular fungal phyla were represented by more than by one order of magnitude more. When looking at the overlap of species level taxa, altogether we found 1.5 million ITS2 OTUs across all those 3 datasets. But to our surprise, there was very small overlap. Here, those numbers in these Venn diagrams represent percentages of unique fractions and shared fractions. So only about 7% of all data was shared between 2 or more data sets. When getting to the level of genera, there were some very strange things happening, as we saw that, for example, the genus Lacaria had around 1,000 OTUs in UNITE and GSM datasets combined, but there were about 35,000 or 34,000 unique OTUs in the global fungi datasets. In the case of genus Piptocephalus, it was a bit opposite. GSM had the best representation of this group with quite reasonable overlap with UNITE data, but very poor representation in the global fungi dataset. We looked into more details about this, and some strange patterns emerged. It seems that some of the data in global fungi may be potentially of very low quality, which generates this these enormous loads of OTUs for certain genera that are well, normally considered to be quite low diversity. So in this case, it's a bubble. And other inconsistencies we can explain by primer biases and also by length biases, because backbio sequencing is not very sensitive to sequencing length, whereas Illumina can sequence only fragments of up to 550 bases in paired end mode. So many of the fungal taxa with particularly long ITS1 or ITS2 subregions might be fully lost. For example, species of Cantharellus, and then many groups of Polytes. So to the second topic now, we were thinking quite hard what to do with all this data. So we decided to address the patterns of fungal endemicity and biodiversity hotspots globally. In this figure, you can see all the samples combined from multiple different data sets representing the full ITS region. So these data include part of the global soil microbiome project. We include actually less than 3,000 plots because several of the plots or samples are collecting from eucalypt plantations, and we are not going to mess this up with real endemicity patterns. In addition, we have 4 other quite big data sets with between 70 sites and 318 sites. In our lab, we sequenced them all for the full ITS region using the very same protocol. And in addition to these third generation sequencing data, we also added data from Sanger sequencing, which has been added to INST and UNITE databases. We included only data for which we knew that it came from soil or soil inhabiting mushrooms, and for which we had quite clear coordinates available. So this subset included around 15% of all the data deposited, which is about 180,000 reads, and these came from roughly 8,000 localities. Again, we started with bioinformatics analysis. We merged all these data into the same taxonomic system, and altogether there was around 1 million fungal species level taxa. We split these taxa into ectomycorrhizal fungi, litter and soil saprotrophic agaricomycetes that are quite well-recognized groups that may warrant conservation. We also took various groups of pathogens, opportunistic human pathogens, yeasts, and other unicellular forms as separate testable groups. So the hypothesis is that larger fungi and especially biotrophic fungi have relatively stronger endemicity patterns compared to free-living and especially unicellular groups. Besides endemicity and biodiversity hotspot determination, we also aim to re-establish the climatic, edaphic, floristic, and anthropogenic drivers of fungal richness and composition globally. So what we have done so far? Not too much. We decided to rely on our analysis on the ecoregion concept, but unfortunately there are too many ecoregions separated globally. So based on vegetation composition and geographical proximity, we decided to pool many of the original ecoregions to generate so-called super ecoregions. So there are about 160 or 170 of such super ecoregions, which will be the basis of this global endemicity analysis. So we restricted the sample size to 40 plots per this super ecoregion, plus included all Sanger sequences to exclude the possibility that a relatively large sampling size, especially in Estonia and Latvia, might affect the results. We are trying a number of different endemicity metrics for this area-based design. First, it's a number of unique species per area, proportion of unique species per area, CRISPs, weighted endemicity index, and potentially also phylogeny-aware metrics, but these may be very difficult to apply without building actual phylogenies. And we will also try network approach and seeing whether partitioning beta diversity into the nestedness and turnover components might give some good solutions. So based on this, we consider building some kind of a super index for endemicity or hotspot values. On top of this, we are going to study how endemicity and hotspots are related to human influence, including land cover change, climate change, and projected future climate change. We will compare these to known plant-animal patterns, and all these different analysis will give some kind of applied output for establishing conservation priorities from the fungal perspective. Just 2 days ago, I got this interesting map from Alex Ziska, our collaborator, main collaborator in this endemicity project. And here, the lighter the colors, the more unique species in terms of proportion these particular super ecoregions have. So you can instantly see that the worst case is New Caledonia, followed by the Western Amazon. These regions, of course, also stand out as particularly high endemicity for plants and animals, especially New Caledonia, because of its more than 30 million years of complete isolation. And we have here one problem that needs quick solving. So we are looking for a person who volunteers generating a specific GIS layer for these super ecoregions. The problematic areas are shown in the gray, where many of the regions are actually split either by geographical coordinates or by the borders of countries for the ease of analysis and equal splitting of data. So with this, I prefer to conclude this presentation, and I warmly thank all contributors of samples, including those directly contributing to this GSM project. Also teams of Martin Zobel, Ülo Mander, Fernando Maestre, and Manuel Delgado Paguerizo for their specific extra data sets. Our core team for this data set analysis includes also Vladimir Mikryukov and Sten Anslan, and endemicity project is led by Alex Seska, Alex Antonelli, Nadia Sotsilovskaya, Nilufar Haktust, and Muhammad Bahram. And I also use opportunity to thank the funders as well. So in case you have any questions, you are most welcome to ask. So I stop sharing this presentation. Mm-hmm. Okay, anybody speak up? I heard some noise, but I couldn't understand anything. Okay, Syria, you have hand up.
Yes, hello, sorry. I have a question about the bioinformatics part. In the first part of your presentation, you mentioned that you clustered at 98% similarity. Is that common for fungi, or is there some other explanation for this?
In my experience, it's the best compromise between splitting and lumping of different species across all various fungal phyla.
Okay, but in comparison to other eukaryotes, do you think for other eukaryotes it would work as well, or would you think it's more suitable for fungi?
I think, well, I know you are interested in specific groups of algae, so you should check what are the limits of these algal species. And I guess one of the best ways for this is to use this UNITE database and this gradual species hypothesis.
Okay. Yeah.
I suppose it will also strongly depend on the group you're looking at. It might be 98% for the dinoflagellates, but it might be around 90% for foramifera, for example.
But then, for example, if I would like to compare different groups to have like an overview of the entire sample, what we could detect, would it be then good to use different clustering levels for different groups?
Of course, especially if this is based on expert opinion.
Okay, thank you very much.
Okay, I saw Brendan's hands.
Hi, yeah, thanks for so much work so far. I'm somewhat on the same line as Sadie's question. Is the OTU clustering based on just the ITS region, or is it also including the V9 region?
The clustering was only based on the ITS region, with all the flanking conservative regions excluded, because that's the most sensitive in this case.
Yeah. Great, thank you.
Hello.
Hi, can you hear me?
Yes, you are loud.
Okay, yes, thanks for the presentation. I have one question. Since you showed us that the difference between the databases for ITS region is huge, do you recommend to use all three databases for also smaller scale studies?
What do you mean by smaller scale studies?
Well, like a smaller scale, like for Estonia, for example.
Oh, I think that this GSM has enough data for Estonia, for example.
Okay,
It
so
very strongly depends on your question.
okay, yeah, so probably it also depends on the region that you are studying, right?
Yes, it's because that the full ITS performs far better compared to ITS2, and in this GSM dataset, all samples are collected in the same way and processed in the same way. So there are no sampling and other analytical biases, as you can see in any other data sets.
I see. Yeah. Okay. Thanks.
Okay. Brendan, do you have a second hand up?
No, I just didn't take it down the first time.
Okay. So Sunil, please.
Hi, Leho. So my question is, you see that there is a difference in terms of microbial population when you amplified with the full length sequencing, and then previously when you amplified with the ITS1 sequencing or ITS2 region only. So does it related to amplification biases related to the PCR, because the full length might, or small stretches could be amplified successfully?
This technical bias is certainly when we compare different data sets, but the comparison within this GSM was related to the very same data. We just extracted either full ITS or ITS2 from the very same sequences. So in this case, it is certainly this kind of identification bias, because when we must use some kind of similarity or e-value threshold to trust whether the sequence belongs to certain taxonomic level, and we cannot be that certain about this when we use ITS2 data alone.
Yeah, because we were just discussing almost on the similar line yesterday when we were talking about amplifying the long read. Then we thought like this PCR amplification could also be an important issue when we want to amplify 3000 or 4000 base pair long stretch.
If you have so long amplicons, you have to have very good DNA. And with longer amplicons, all those PCR biases related to differences in fragment length among different organisms becomes very much emphasized.
Yes. Okay, thank you.
So, Jakob.
Yeah, I want to switch topic a little bit. So it's for the biogeographical analysis. I think there's possibilities to go both from like a top-down approach where you start with existing definitions of zones, or you could build it up from the bottom up and see how the individual sampling points are similar and not. And I mean, since nobody have produced a map of fungal biogeographical zones before, I think both approaches could be fine. But it seems that you're mainly following trying to work with existing zones, or did I get it wrong?
Yes, well, we are working on the existing zones also to see whether these match with those regions of plants and animals. And later, or in parallel actually, we are also running an approach where we are using more delimited sampling units, for example, based on grids or individual plots. So based on these data, we can this can complement our understanding of this.
Great. Yeah, that sounds good. I think it's really nice to go both ways and see what is coming out.
Sure. Yeah. Hi, Adrana.
Hi, Leho. You maybe explained this, but I was curious. How are you guys correcting for sampling bias per ecoregion to calculate species richness, for example?
You mean sampling bias by the different number of plots per region, right?
Exactly. Yes.
Mm-hmm. Yes. Well, that's a question we are handling right now. So I will see. We will certainly have to account for the number of sequences or the number of plots as some kind of covariates, or initially produce some kind of residuals based on the sampling depth to proceed. So we will see which performs best. So this is what we are actually starting right now.
Okay, perfect. Thank you.
So I will also inform you that within a few days or so, you will also get access to the preliminary manuscript. Please, again, those who have not added their affiliations or contact, please do so. And there are also a number of people who I haven't been communicating with directly. So these people should explain their contribution to me. And thereafter, I will also collect some expressions of interest if certain people want to do some additional analysis based on geographically more restricted data sets. So Roberto, please, I see your hand.
Hi, Leho. Good morning, Mexico. As you know, the OTU threshold will influence the distribution of species. So for endemicity, have you think on working on different thresholds to see how much the distribution changes? I have done so, and sometimes you find a threshold where the distribution stop growing, so that also could be informative.
I certainly agree, but as you see, the dataset has like more than 1 million species, and when you increase this threshold to 99%, you will get 2 million species. So it is even now a huge burden to computers and even computing centers to handle these data. You even can't visualize it all in a single Excel table, for example. So although I agree with your approach, but I regret to tell that we are not probably going to try those different levels of thresholds.
Okay, thank you.
Kadri, please.
Do these current unite species hypotheses include representative sequences from all your global sample or just part of them?
Here we excluded those other back biosequences from the UNITE and INST reference dataset. So we are not comparing the same samples against these same samples.
But I actually meant that if I open the SH view, are these all now incorporated there, or some are yet to be incorporated?
We have incorporated the Estonian subset, which was published in summer last year. And we have incorporated some of the very long read data. These had a small amount of sequences, but we could use them for identification in this case.
Okay, so actually majority is not yet in there. Yeah.
No, almost nothing from this set is there. Because also the Estonian samples, much of these we resequenced using this sequel2 method to get deeper sequencing data.
Okay, is there a plan to incorporate in near future those or part of those?
Yes, together with Kessi and Urmas, we are quite actively working on this, especially how to do it in the best possible way so that you can still visualize your species and then your computer doesn't crash, because it would mean like more than 10-fold increasing the entire UNITE dataset.
Yeah, I can imagine. And okay, very good. Thanks.
Saleh, please.
Yes, when you are going to sequence the full ITS region, how do you go around the 5-point-S region? Are you going to exclude it or join these two regions, or you are going to analyze the ITS1 and ITS2 in a separate data set? What is your approach to do
it? We are not going to do anything. We already did. We already included the entire full ITS, which includes ITS1, 5.8S, and ITS2 in one single stretch. Okay, Any other questions?
Hello,
Hello, can you hear me?
Hello, hello.
I'm Marika. I say hi to everybody. I just want to know, what are your future plans?
Future plans. How long future do you mean?
Well, since the amount of data you have is probably the next 100 years. No, I'm not kidding, but what are you going to do? Are you going to include more sampling, or are you going to keep on mining the data that you have?
No, I'm not going to focus on this same data set for long. I predict that we are going to get this database paper out, and we are all together getting this endemicity paper out. And already when the data set is out, then other researchers, including you, can postulate your own hypothesis and access this data to answer your own questions. But after these two papers have been achieved, I will more focus on extra global data that we have obtained from plant leaves, water and sediments, dust, and in more distant future, trying to bring all these global data sets together.
Thank you.
Saleh, you still have hand up?
No, actually, I asked my own question. That's it.
Okay, yeah. You are just keeping your hand up. Okay, so I think in this case we are going to finish this session. I thank you all very much for joining. Very soon I will share this link to the manuscript, and in the forthcoming mail I will also add some additional points or questions that you might consider. So thank you very much, and enjoy the day, morning, evening, or night, whatever you have. And this presentation will be also uploaded, so those colleagues who couldn't see or who want to revisit it because of poor connection can safely do it. So you will get the link. So, bye.
Bye.
And thanks.
Bye-bye, thanks.